Medical genomics acquires and analyzes information pertaining to individual patient genomes with the purpose of preventing, treating, and curing diseases. It is truly a multidisciplinary field, and it includes genomics, biochemistry, population genetics, statistics, computer science, and clinical research. The Center for Medical Genomics at Penn State was founded in 2008, and over the years, the center has grown and became one of the most successful collaborative efforts linking both the University Park and College of Medicine campus. By collaborating with the College of Medicine, we can put our scientific questions in the clinical context while acquiring data on large patient cohorts. In collaboration with Dr. Ian Paul from the Department of Pediatrics at the College of Medicine, we are studying the causes of childhood obesity from the perspectives of genomics, microbiomics, and epigenomics.
25% of 2 to 5-year-old children in the United States are already overweight or obese, so preventing that is very important. We've been fortunate to show in our randomized clinical trial that we can prevent rapid infant weight gain in the first year after birth and have babies be leaner at age 1 year when they receive our intervention. With the help of our biologic collaborators up at University Park, we've been able to determine that those babies who have rapid infant weight gain have a reduced diversity of their gut microbiome, and that potentially is a very important finding. This multidisciplinary collaboration is key. Adding the genetic piece to the puzzle is critical so that we can really have a very comprehensive description of our patients and help us determine which interventions work for which children.
Collaborations are central to function of the center. We enjoy and we build upon interacting with scientists with complementary expertise and skills. For us, this happens naturally.
I'm a statistician. I was trained as a statistician, and in fact I never really studied anything related to biology or medical research except maybe in high school. The center helped hugely because the computational and statistical aspects are intertwined into the biomedical research from the very get-go. And this comes about because we work together in a very integrated way in the center.
To study mitochondrial diseases and to investigate whether their occurrence is influenced by maternal age, we are studying the transmission of mitochondrial DNA mutations from the mothers to the children.
My role in this project is to bring skills and competency in methodology that has to do with mining large data sets for patterns, as well as, where possible, trying to model the mechanisms that underlie the data.
In my group, we are sequencing and analyzing the mitochondrial DNA in families. We're using the methods that allow us to look at mutations present in as low as only 1% of mitochondrial DNA.
With the data produced, we were able to produce estimates of the germline mitochondrial DNA bottleneck as well as mutation rate. Another very important thing that we were able to do was to investigate the association between the prevalence of heteroplasmies in a child and the age of the mother. This type of insights actually can only come about by this close intertwining of computation, data analysis into the biomedical research. This type of interdisciplinarity is really a hallmark of the center.
Medical genomics uses both computational and experimental approaches. Based on the data collected in our and other laboratories, we can build computational models that can be further tested in subsequent wet lab experiments and also in clinical trials.
With computer science, we can analyze the whole genome the same time as it would take by hand to analyze just one gene. But where it's really strong, especially together with statistics, is to find patterns in the data that we get that are not discernible to the human eye.
My laboratory's previous research was focused on understanding the biochemistry of microsatellite DNA replication through a phenomenon known as microsatellite instability in cancers. This instability had been described in tumors at only a dozen or so microsatellites. When we began our research with the CMG through an NIH-funded grant, we were able to greatly expand the scope of our studies to interrogate millions of microsatellites throughout the human genome.
Computation lets us analyze mutation across whole genomes of hundreds of samples. The patterns of instability we're looking for are subtle, so it's important to have this much data.
Through our research, we found about 1 million microsatellites that are changed, and many of these were changed in specific human populations. In the near future, we would like to examine mutations that are arising in precancerous tissues of patients who are at risk for cancer. Our collaborations with colleagues at the center are vital for this ongoing research.
Graduate students and postdocs are central to all of our projects. By working with professors specializing in different disciplines, our students are acquiring interdisciplinary training. They also learn how to communicate their findings to scientists specializing in different disciplines. This makes them well prepared for their future careers.